This technique leverages multiphoton laser scanning microscopy in conjunction with laser flash photolysis of a photoactivatable nicotine molecule, allowing precise activation of nicotinic cholinergic receptors.
This technique allows for fine spatial temporal control over nicotinic receptor agonist application, providing a powerful tool for the study of receptor functional expression patterns in cholinergic, synaptic, or volume transmission.
Before beginning the procedure, use a programmable pipette puller to pull a glass micropipette with a 20 to 40 micrometer opening diameter. Strain approximately 1 milliliter of recording solution through a 0.22 micron filter, and resuspend enough lyophilized photoactivatable nicotine in the filtered recording solution to yield a 2 millimolar final concentration. Backfill the pulled local application micropipette with 50 microliters of the photoactivatable drug, and secure the pipette in a pipette holder mounted on a micromanipulator. Connect the pipette holder via an appropriate length of tubing to a pressure ejection system capable of sustained low-pressure application, and use the micromanipulator to maneuver the local application pipette into the extracellular recording solution. Position the pipette tip slightly above a mouse brain tissue sample approximately 50 micrometers from the cell of interest, and briefly apply 1 to 2 pounds per square inch of pressure to the pipette. There should be minimal to no displacement of the cell of interest.